Alright, the next part of the video lecture series for Chapter 9 is going to be looking at the physiology. But because we've worked our way to the microscopic structures of skeletal muscle cells, we'll actually be starting with the third step in muscle contraction, which is a sliding filament model of contraction. So before we get there, I just want to give you an overview of everything that's going on to get us to that point. I'm going to try to use paint to illustrate what's happening, so this will be a really rough overview. There'll be probably pretty crude drawings as far as accurate, but just pay attention to what's happening. So we're going to start with the brain. So we'll use a circle as a horrible circle. Let's undo that. That's better. Alright, so we're going to start with the brain. The brain has, leaving from it, let's make this thicker, a motor neuron. So here's our motor neuron. We're going to say that it's kind of bent. Alright, now it's really fancy. So from the brain, there is a motor neuron with signals that are leaving. So that means that this motor neuron is sending an efferent or output signal to something. And the efferent or output signal in this case is going to go to a skeletal muscle cell. So let's just give the end of our motor neuron some branch structures. These branch structures are actually called axon terminals because it's at the end of a long process from a neuron, which is called an axon. So at the end, it's the terminal part of the neuron, so axon terminals. Alright, so again, we have the brain. The brain has a motor neuron, which is of the somatic nervous system. And let's make that viewable. Going to a skeletal muscle fiber. So let's draw our skeletal muscle fiber. Let's make him red. And then let's say that our sarcolemma, so our plasma membrane, is going to be in black, just so we can tell the difference. Okay. And let's just move the whole thing over a little bit. Alright, that's good. Alright, so we have the brain sending an output signal to a skeletal muscle fiber. Not bad. This is our step up. Alright, this location where the end of the motor neuron gets really close to the skeletal muscle fiber, this location is called the neuromuscular junction. So I'm going to abbreviate that. Neuro, N, muscular, M, J, junction. And so the name's just telling you this is a location where the neuron and the muscle get really close together. And again, I'm abbreviating, I'm shortening, we're going to go into more detail. But what's happening at the neuromuscular junction is an electrical signal that the neuron had. Let's make that thicker. But an electrical signal that you can find in the neuron is going to jump from the neuron to the sarcolemma. And remember what the sarcolemma is attached to. The sarcolemma is attached to T-tubules. So that electrical signal is going to jump into or onto the sarcolemma, and it's going to travel along the sarcolemma until it reaches a T-tubule. Once it reaches a T-tubule, it's going to be able to travel inside the cell, which is great because we need the T-tubule to tell the sarcoplasmic reticulum, that's what I'm drawing here in blue. Let's do another one. Remember, it would be kind of like a web-like structure out here, but we're looking at a triad where the blue terminal cisternae of the endoplasmic reticulum, or sarcoplasmic reticulum for a muscle cell, are touching the green T-tubule. Alright, so what we have is the brain sends an output signal via a motor neuron to a skeletal muscle fiber. The location where the electrical signal from the motor neuron moves to the sarcolemma is the neuromuscular junction. The electrical signal, once given to the sarcolemma, is going to travel along the sarcolemma to a T-tubule, which is continuous with the sarcolemma. Then the electrical signal is going to travel into the cell. And as it's traveling into the cell, it touches the blue sarcoplasmic reticulum. Now there is a protein that attaches the blue sarcoplasmic reticulum to the green T-tubule. And when electricity goes past this protein, it opens. When it opens a channel, and what happens is a bunch of calcium, I just Ca2+, a bunch of calcium flows out of the sarcoplasmic reticulum. So specifically, we're looking at calcium flowing out of the sarcoplasmic reticulum. And that means that now there's going to be a bunch of calcium in the cytoplasm of the muscle cell. So it's going to be a bunch of calcium in the sarcoplasm. And calcium is the final go signal for contraction. Alright, so what we just looked at, where the electrical signal goes down the T-tubule and causes the sarcoplasmic reticulum to release calcium, that is called excitation, contraction, coupling. Because what we did is we linked the electrical signal, the excitation, to calcium, which is the final go signal for contraction. So we linked electrical signal to contraction. So excitation, contraction, coupling, or the ECC. Alright, the next step is even more microscopic than what we've been looking at. Instead, we're going to be looking at a myofibril. Let's make that pink. And you'd have a bunch of these myofibrils just hanging out inside the cell. And when calcium gets released, calcium is now present to bind to something. Before there was no calcium being released, it wasn't inside the cell, therefore it wasn't binding to stuff, it wasn't doing anything. But I said calcium is the final go signal for contraction, so it must do something. Its release can't be its last step. And it does do something. So let's take one of those myofibrils and take a closer look at it. So let me draw or get that myofibril out here. So again, it's happening inside the cell. I'm just pulling a myofibril out so we can get a better view. Alright, so here's my myofibril. I know that inside my myofibril would be, let's do this, a thick filament called myosin, and a thin filament called actin. Let's just use a line for that. And there'd be a lot of thick and thin filaments. It wouldn't just be these two, so we're simplifying. And on the myosin, there would be globular heads. And these globular heads want to bind to actin. Because on actin, there's actually an active site that wants to bind to myosin. So myosin has a binding site for ATP to power this process, but after it's used ATP, the myosin head would like to bind to actin. But normally, troponin and tropomyosin, which are two proteins, are in the way. So you'd want to move those out of the way. Let me actually pull up the PowerPoint so we can go back and look at these structures. So here's myosin, the thick filament. It has globular heads. We like to put these subunits together to get a really thick filament. And the globular heads would bind to ATP to power this reaction, and then they'd want to bind to actin. They want to bind to this dark circle right here. This is the active site on actin, and that's where myosin wants to bind. But right now, troponin and tropomyosin are in the way, so this rope-like structure and this globular structure are in the way. But calcium actually binds to troponin, and when calcium binds to troponin, it twists. And when troponin twists, it moves tropomyosin out of the way. And if tropomyosin is not in the way, then myosin, this molecule, can bind to this active site. So calcium is really important because it moves the troponin-tropomyosin cascade out of the way. So we have the neuromuscular junction, where the electricity jumps to the sarcoplasm. The electricity allows for calcium release when the electricity goes through the T-tubule and tells the sarcoplasmic reticulum to release calcium. Calcium then binds to troponin, which is around actin, moves troponin and tropomyosin out of the way, and now, because calcium bound to troponin, myosin can bind to the actin active site. And when troponin is out of the way and myosin binds to the active site on actin, now they're touching each other and the myosin head pivots. It kind of swivels. And this swivel reaction allows for more overlap of the muscle. It allows for overlap of actin and myosin. And that overlap, let's go forward just a minute. Please ignore the multiple slides that are passing by your screen. Here we go. So what's happening to the sarcomere when actin and myosin can touch each other? We get shortening. So here we don't have as much overlap. There's spaces where we have just actin, that's our I band. There's spaces where, even in the A band, where there's already some overlap, there's spaces where we don't have any actin, right in the middle, where the H band is. But when a myofibril contracts, all the sarcomeres shorten, and that means that we're getting more overlap. So we don't have areas with just actin, we don't have areas in the A band with just the thick filament. So we get more overlap, not shortening of the fibers, they're just overlapping more. And this process is only possible because myosin touched actin. And myosin was able to touch actin because calcium was released and moved the troponin-tropomyosin cascade out of the way. And when myosin touches actin, that is called a cross bridge. Usually cross bridge cycle because there's steps that repeat. Okay. So the 3 steps for skeletal muscle contraction. Skeletal muscle are the neuromuscular junction, abbreviated NMJ. Step 2, excitation, contraction, coupling, abbreviation ECC. And third, we have the cross bridge cycle. So I know this paint drawing is a little bit all over the place, but also make sure you're watching the 3 videos that are shown in the upcoming physiology for muscle, and that you're watching the videos repeatedly and then going back and looking at your PowerPoint and seeing how everything matches up. So again, if you don't know the anatomy, start there, then watch this kind of brief overview, and then make sure you're following the video lecture that goes through the physiology in the PowerPoint. All right, 3 steps for skeletal muscle contraction: neuromuscular junction, electricity must go from neuron to muscle. Excitation, contraction, coupling. The electricity has to lead to contraction, which requires the release of calcium from the sarcoplasmic reticulum. And then cross-bridge cycle, which you can abbreviate CBC. You have to have myosin bind to actin in order for a sarcomere to shorten. And sarcomere shortening leads to muscle cell shortening leads to the muscle shortening, and that leads to muscle contraction, or overall muscle contraction if we're looking at muscle as an organ. Alright, so this is rough. I told you the drawings would be crude, but you get a general overview of what's happening. We're going to be talking about motor neurons of the somatic nervous system, and we're talking about skeletal muscle and how exactly that's allowing for muscle contraction. And it all leads to that cross-bridge cycle. Alright. So I'll go back, and for the next video, we're going to start with the sliding model of contraction. So that's what we'll be looking at.